All right, here we go. Danny, do you want to do the introduction?
Sure. Oh, okay. So I guess we're gonna start by introducing the founders. So my name is Danielle Choi. I'm currently a sophomore, and I'm very interested in neurodegenerative diseases and neurology.
Yay! And then we'll have Gerline next.
Hi, my name is Gerline. Um, I'm one of the co-founders, and I'm really interested in structural neuroplasticity. And I think one of the main things I see for BiouBrain is its ability to reach a lot of people and be an avenue of access for a lot of younger students, especially at a time where there's not much out there.
My name is Jolyn. I'm a junior in high school. Um, I'm just really excited about neuroscience in general, but specifically, um, I do like neurodegenerative diseases, and I also am interested in like the genetics or the genetic level, I guess, of diseases in the brain or in the nervous system. And I guess for BioBrain, I'm also really excited to just like see other people get really into neuroscience, and I hope that we can serve as a nice place for resources. Yay!
Yay!
Um, So I guess just a couple of things. So like what is BioBrain? So it's a student-led initiative focused on helping bring neuroscience-related resources to students in both elementary and secondary school. And our mission statement is to make neuroscience more accessible to younger generations. And some of the main projects we've been working on is like the Neuronic comic, which is like a comic series to help make neuroscience much more fun and engaging. Also Neurogram, which will probably be like a database of like digital diaries related to neuroscience, as well as Brain Hub, which will be like a series of free educational games that teach a little bit more about the phenomena of the human brain. And so our big project that we're here today for is Neuronicbooknook. And it's like a bi-monthly journal club where neuroscience lovers gather together to discuss recent discoveries and publications in the field. And yeah, the only requirement is to be interested.
Sounds awesome. Then I guess we can get started then. Looks like we have Girlene's paper first on the agenda today. Girlene, what's your presentation gonna be about?
The presentation is going to be about a very recent article. It was published in Nat Neuro, and the title was "In vivo hyperphosphorylation of tau is associated with synaptic loss and behavioral abnormalities in the absence of tau seeds." So I am excited to tell you about that.
Yay! Do you want to share your screen for the presentation?
Yes, please. Let me just figure that out.
Didn't go with it.
Okay. Sharing. Okay, I'm gonna send you a request.
All right.
Okay. Just 1 second. Let me get it open. Okay. Okay. Let's get to it. Okay. Can everybody see?
Yes.
All right, thank you. I'm gonna get started now. Okay, so this is the article. And published in Nature Neuroscience in 2024. And authors were What's More and Okay, so the background and significance. So, neurodegeneration is a very important area of interest right now in research for neuroscience. And right now, one of those proteins that we're looking at that might be like a huge cause for is the tau protein. And like, especially like tauopathy was just coined itself as a separate term because of like its like significant impact in like causing neurodegeneration. It is like the aggregation of tau causing progressive neurodegeneration. And basically, what we've seen is that a lot of like aggregation of certain like proteins, related protein is like amyloid beta, for example. The aggregation of these proteins is like suspected to be a huge cause of like neurodegeneration as a result. And essentially, hyperphosphorylation of tau occurs as a result of the MAPT gene, and these mutations in the MAPT gene are seen in neurodegenerative condition, even when pathology is sort of spaced out or like isn't exactly like going by a pattern. And the tau is observed to be associated with the snare complex and vesicle exocytosis associated proteins, so it's like very important in like presynaptic molecular mechanisms and can be transported from presynaptic to postsynaptic neuron as a result of neural activity via secretion. And because of this, like tauopathy spreads very, very fast in the brain. And it was seen that like synapse loss, which was at significant rates, result in hyperexcitability of neurons, which means they're active all the time. And like this causes like cell death as a result of metabolic exhaustion. And when the cell death happens, it causes neurodegeneration down the line. And essentially, where this is important, is this article that ties it all together. Okay, so the authors of the article were Naoto Watamura and Martha S. Ferrani. And these were the first article authors of this article, and the supervisors um, who helped edit and conceptualize were Duff and Saito. Other editors include Bordanis, Camprese, Brinkbaum, Santabrogio, Vendrasco, Zetterberg, Zetterberg did declare conflict of interest for this article, but and these editors also helped like conceptualize a little bit of methodology as well. And I think the remaining authors helped edit or contribute to methodology from various labs as well. So yeah, this was a huge project. It was very large scale and it was very rigorous. I love it. So the hypothesis for this article was that introducing the specific MAPT S305N and frontotemporal causing mutations into the humanized MAPT genome with previously knocked-in tau will affect the phenotypic expression of the tauopathy and as a result of hyperphosphorylation in mice when controlling for conditions such as habitat, hunger, and risk of predation in the mice as a result of behavioral tests. And related articles include the following. The bottom one is actually something related to what Fawani has done before with a few co-authors actually. And so they do tend to work on things like this in their lab. I love it. Okay, so I'm just gonna go through the experimental design and methodology really, really quickly. Um, so um, so essentially at first they had some genome editing. They added in the mutation essentially using a base editor and uh, after which they basically they allowed the like sort of cells to be injected into the mouse zygotes, so then that would help the like genes sort of like that would help the genes come together, and they had raised the mice essentially that came from that. And once the mice were at an appropriate age, they sacrificed them. They conducted some tissue analysis using some Western blot and Gauss silver staining, and they were also actually given some human tissue of like that was showing tauopathy as well. And so they analyzed that and looked at it side by side as well. And additionally, what they did was they also did some synapse counts for the mice tissue, as well as like some behavioral tests to really show the pipeline from synaptopathy to neurodegeneration, as like especially looking at like frontotemporal dementia, which was what the behavioral tests kind of like really focused on trying to um, bring out. Okay. So like because of this, they saw that like prion formation as a result of the scans, it wasn't really seen in the mutation model mice. And it was shown that like the mutated like tau wasn't really able to form prions. And however, there was still like frontotemporal dementia symptoms and like signs shown in the mice. So it kind of went to show that in these stages, it's not necessary for the mice to like be exposed to like prions in order for dementia to onset in the frontotemporal regions. So, however, behavioral deficits were seen along with considerable synapse loss, which kind of went to show that synapse loss might be something to look at rather than prion formation in this model, especially. And I just want to show a few scans of the synapse counts here. As you can see, the gray. Wait, can you guys see my cursor?
Yes.
Okay. Okay. I'm sorry. I don't know how to do the thing the thingamabobber. So I'm gonna have you guys with the cursor instead. Okay, so as you can see here, these are the synapse counts for the wild type mice quantified. And then this was the initial, the pink was the initial like Mackey mutation, and then these were the specific mutations that we were looking at in the purple and green blue. And you can see that in these like areas, the synapse count is really considerably like lower in the mutation that we were looking at. Like there was a drastic difference compared to all the other like mutated mice. However, like the silver staining kind of went to show like that the thingamabobber. Give me 1 second, please. However, the silver staining went to show that it was actually the opposite, like in terms of the mutation versus the previous mutations that we were looking at. So that's an interesting thing to see. Okay, so I think what this leads us to was that like hyperphosphorylation, like as a result, causes tau to disconnect from the microtubules and relocate away from the axon, which like kind of like disrupts the like secretion of tau upon axon activity, as it was previously mentioned that it was related to the snare complex. And this relocation kind of like reduces the expression of AMPA and MDA receptors in the spine, which actually drastically affects neuron function. And this like because there's less like receptors, it causes the activity to slow down, which causes the dendritic spine to reduce. And as a result of this dendritic sparsification, this excitability happens, which essentially leads to metabolic exhaustion, cell death, neurodegeneration. And yeah, oopsies. Sorry about that. And humanization of MAPT is a main cause of behavioral alterations that were consistent with the phenotypic expression of frontotemporal dementia. However, it doesn't really go according to what would normally be seen as a result of dendritic sparsification, and like further investigation is needed. So future directions and clinical context for this article. This was a like brand new model that they were looking at, and seeing this model like accurately like sort of show um, neurodegeneration as a result of tauopathy, it like opened up big doors. It kind of like allows us to look at the underlying like molecular mechanisms that sort of initiate this tauopathy, like to cause neurodegeneration, which tells us a lot. Um, and this kind of brings on like potential, like where can we like, look for places for pharmacological treatments or like what avenues can we look at? And then the protein-protein associations of tau are really what we're trying to look at and how they might play roles in aggregation and like more so than like prion formation. As we saw in this model, prion formation did not happen, but there was still neurodegeneration. This, however, like prion formation does happen in the late stages of a lot of mice models. And so in here, because it doesn't happen, this can like really only at the moment be used as like an appropriate model of early stage tauopathy rather than late stage. So that is something to look at. However, like future investigation is needed, and I think this is great. So these are my references. The top reference is the article that we were talking about. These are in Nature format. Thank you so much.
Oh my God, yay! This was so exciting. Actually, if you don't mind, could I ask a couple questions?
Of
I think
course.
I might have missed this, but are you saying that like is it because of the telepathy that like the synapse count was very low?
Yes, I think so. I think because the tau it got relocated from the like snare complex area to the soma of the neuron, the like receptor activity or the receptor expression reduced, which essentially like led to dendritic sparsification because there was not much activity, so the spines kind of went down, and so there weren't that many
Uh,
synapses. And then because there's not that many synapses, it's getting like overactive, the neurons. They're like kind of working too much. And when they get overworked, they die.
did I? Okay, that makes a lot more sense. Also,
That was a great question.
I was trying to like listen. Sorry, I was trying to listen, but like I think my brain was just moving a little bit too slow. I kind of missed the part about like the hyperphosphorylation part about tau. Would it be possible if you could go over that again?
So I think the hyperphosphorylation of tau relates to like a structural change in the tau. So this is where the protein makeup of tau essentially because of the genetic mutations, it gets like too much phos
Phosphate, too much
phosphate,
phosphate.
yeah, phosphate into it. And because of this, it's like well, it's like mechanisms are also like going to change, but like here because it's like chemical properties, what it really does is it disconnects from the snare complex area that it needs to be at and instead goes into the soma and it stops doing its job.
Whoa, that's kind of crazy. Um, yeah, I'm really excited to see where this goes. I mean, like, I think because we know in Alzheimer's it's like tau tangles and like the beta amyloid plaques. Like, I'm really, yeah, curious to see like where this is gonna go in the future
Yeah.
because it's like kind of a new like thing because people are mostly like looking at like beta amyloid plaques and like the APP like precursor protein in Alzheimer's. So,
Yeah, that's absolutely right. And to think that like now, like it might be suspected that tau like prime formation isn't even like the start to it all. It's like
mm,
really, it's like something. And honestly, I think it might bring in more light to like synaptopathy and how it will relate to neurodegeneration and exactly how many causes of there like that there are for synaptopathy to happen. It
so
was a
would you say
great
like it would
question.
be worth like investigating if like tauopathy is kind of like a precursor to prion formation?
Yes, I think at this point it is suspected to be like a precursor to prion formation. Except in this mutation, there was no prion formation for some reason.
Yeah.
And instead of like the tau like kind of, like forming the prion and harming the neuron in that way, it kind of showed that like the prion, it really like it didn't matter. There was neurodegeneration either way. So I think at this point, like it might be important to look at like how prions in themselves like affect the connections that neurons have between themselves and how like maybe possibly a deprivation of these synapses might lead to neurodegeneration in that case as well.
Wow. I think those are all my questions. Danny, do you have anything?
I guess with like Alzheimer's, I know there's like so many different causes. Like it's not just like the plaques and the tangles. Do you think like the mutations are like somehow related to like epigenetics where the environment kind of turns on or turns off the genes?
That's a great question. I think to some degree, yes. I was actually like listening to a Neurosci 101 lecture a couple years ago by a professor named Dr. Ju, who had talked about like a lot of like epigenetics and how it could relate to neurodegeneration. Some of the main points he mentioned, other than like genetic like outcomes and like it being passed on through families, was this compound called 1-octantriol, which was like a compound released by black mold in a lot of places, like old buildings, which like showed that. So essentially, what he'd done was he had like fruit flies, Drosophila, and when exposed to 1-octantriol for a certain amount of time, they showed signs of Parkinsonism, which was like a hallmark of Parkinson's disease. So it's like really something to look at. And there was also some talk about some yeah, he made a really good point about the gut microbiota as well as like I think molecules, prions traveling up the vagus nerve and into the brain, and that being a potential cause for neurodegeneration, such as Alzheimer's disease and Parkinson's as well. So I think it's definitely something to look at. Those are great questions, guys. Thank you. Yes, I'm glad, honestly. I think is neurodegeneration a like area of interest for you too?
Definitely.
We've got exciting things coming. Stay tuned.
Yay! All right, does anyone else have other comments? Or otherwise, I'll move on to my presentation.
I think you should log into your presentation.
Okay, sounds good.
All right. So I'm gonna stop
Thank
sharing. So excited to see
you.
yours.
All right, so my presentation, yay! It's about, hold on, let me see if I can find the spotlight thing. I think there was one here. Oh, here it is. There we go.
Oh
Um,
my God.
isn't that cool? Um, yeah, so my presentation, I looked at this paper about elevated genetic risk for multiple sclerosis, and that a lot of that comes from steppe pastoralist populations. Um, just as like an introduction, so multiple sclerosis, I'm gonna shorten it and just say MS while I'm talking. It's a neuroinflammatory and neurodegenerative and autoimmune disease that affects around 2.8 million people around the world every year. Um, and basically, what it does is that the body starts attacking like the myelin sheath on neurons in the central nervous system. And so basically, like your body just starts like eating away at the myelin sheath. And that's really bad because the myelin sheath is needed to send like signals really fast to other parts of your body. So, um, it's a very interesting and pretty deadly disease, debilitating, and that's why it's significant. Um, about the actual like genetic related information. So what we knew before this study was that MS is linked to immune-related genes, which would make sense because it's an autoimmune disorder. But it's like unknown how this genetic risk came to be. Like, was there positive selection for this like genetic risk? Blah blah blah. And specifically, people were looking at the HLA regions of chromosome 6, where the genes will code for like the proteins in your body that identify whether cells are self versus non-self, and then thus like to attack them or not. So the study is going to look at the HLA regions as well. And just a kind of foreshadowing, Barry et al. Finds that the descendants of steppe pastoralists are at a high risk of MS. And just for a little bit of background, steppe pastoralists were a group of nomadic people. They were living like around modern day like Central Europe, so like around Georgia, Russia, that kind of region. And they ended up just like migrating like all over to Europe. A lot of them are in Northern Europe now, and some downwards to India, even to like Western China. Yes. These people, so the steppe pastoralists, like came mostly during the Neolithic period, like Bronze Age type time. And they brought in a bunch of like lifestyle changes that would contribute to a bunch of positive selection. And yeah, since this was like more of a genetic like analysis than an experiment, I had a moment trying to find the hypothesis, but it was like pastoralist ancestry increases genetic risk of multiple sclerosis due to radical lifestyle change. What they did is that first they matched present day like European genetic, like people with European genetic ancestry, specifically like white British, like self-identifying like white British people. In the UK, they got their genetic information and matched it with medieval and post-medieval genomes. So some of this is old information, some of it is new. And then they found a lot of like single nucleotide like polymorphisms where the like one nucleotide like replaces another in a mutation. And they went to check whether these variants that were associated with multiple sclerosis underwent positive selection, when that happened, where that happened, and the environmental condition. So then after that, they compared the SNPs of the DNA in steppe pastoralists from all that time ago to the people in the UK now. And then, so to confirm their findings, they also conducted 3 types of data analysis, just like regular association analysis, and then 2 association with local ancestry, and 3 haplotype trend regression. All of them just ended up like proving the same thing. And just for more information, they tested backwards up to like 15,000 years ago. And one important thing is that they calculated the LDA, like linkage equilibrium of ancestry, which like measures the correlation between the ancestries at like different mutations, like the SNP is like each nucleotide, to study recombination patterns. So basically they wanted to like see like what happened with the SNPs, like when did they mutate, and then how it got like passed down the genes. So yeah, and most excitingly, the results are in. So the results, what they found from comparing like modern day genetic information with like steppe pastoralist genomes, is that there were really, really, really big differences in chromosome 2, the LCTM-CM6 region, which basically just codes for lactose intolerance or tolerance. And then the part that we're focused on, chromosome 6, the HLA region, where it codes for the human leukocyte-like antigen. And they found a specific SNP that carries a really high risk for MS. And very interestingly, what they found it first appeared in like a northern Italian like individual, like about like 5,800 BCE. But it wasn't until like 500 years later during the emergence of like Yamnaya culture where they actually like started becoming nomads, like fishermen, like when they had that big like change in lifestyle, that was when like this mutation or this like SMP started to get like passed down a lot. So clearly it had to be like used for something very that would be very useful for like their survival. And what they found is that like after comparing all the groups that like kind of existed before, during, after this like time period in that region. So there were a lot of like farmers, nomadic groups, um, and all of that. The researchers found that steppe ancestry, like the steppe pastoralists that we're talking about, have the most out of all of those groups contribution to modern day risk of MS, which is really interesting because that risk had to be, I guess, positively selective throughout all the generations. And basically, what they found is that since they're all in like the HLA region, the mutations during that time were actually like trying to help protect like these nomadic people or like the steppe pastoralists from pathogens and like viruses and a bunch of other things that were probably just going around at that time. But as a byproduct, like due to the mutations, that probably just like increased the risk for MS and it just didn't get negatively like selected against. And people are just more susceptible to MS, like if they have this like steppe pastoralist ancestry. Um, and then about the LDA scores, interestingly, about the LCT and MCM6 region for lactose like tolerance intolerance, it had a very high LDA score, meaning that there was recent like selection going on. So the selection for lactose tolerance intolerance was recent, but for the HLA region, the LDA score was low, suggesting that the selection was A, pretty long ago, and B, was due to mixed ancestry, which makes sense because around that time, like, that was when a lot of people started moving around, including the steppe pastoralists. And I just pulled a couple figures, figure A, B, C, D, E, F, G. These just show like a couple of the different ways they did some analysis, and all of them just show like how much like MS like associated with the HLA region. And they were just trying to show here they like developed their own way of analysis, HTRX, and it does seem to show like MS association the best, or variants in MS association the best. And H is another LDA related piece of data. It's like a plot for the SNPs on LD on chromosome 6. So they just found a lot of these like single nucleotide polymorphism like mutations, and these with like the darkest colors are the most like linked to or connected to MS in modern day people. And oh, I didn't include this in the results, but they also tested for rheumatoid arthritis, which is also like neuroinflammatory and like autoimmune. What they found is that like really interestingly, it was a lot of people also had like RA, related autoimmune like mutations in chromosome 6. So I think that just further goes to their point that chromosome 6 in the mutations in CHR6 contributed a lot to modern day like these autoimmune like disorders and diseases, that type of risk. So just to wrap it up, steppe migrations to Europe, basically at first like when the steppe pastoralists were in Central Europe and then they migrated to like Western Northern Europe, that contributed to modern day like genetic MS risk, and that was like around 5,000 years ago. So what they saw in the genetic information was that like around 500 years ago, the selection for MS risk, that is like when it started to happen. Because before, when it was just like farmers in like Europe, then there wasn't that much like change or the significant change that they were looking for in chromosome 6. And then RA, rheumatoid arthritis, they saw that there was genetic risk coming from other groups other than steppe pastoralists. And what they found in lifestyle, I actually would have loved to see more specification in lifestyle change, which I put in future directions. Um, they talked about there was increased consumption of like domesticated animals, so that probably contributed to like a lot of the diseases running around and thus like positive selection for the genes in chromosome 6 that would have helped protect them at that time, but now like those mutations just end up becoming making these people more prone to MS. And some of the limitations mentioned were that they didn't have that much like evidence for further study because of the vaccination programs nowadays. So there wasn't that much like genetic evidence in people now that they could look for. And also there just wasn't that much genetic evidence from before. And yeah, that's pretty much it about my article. Are there any questions?
Can I ask a question?
Yeah.
So I think you mentioned that the selection pressure for these populations was that it protected against like certain pathogens that were seen in the environments that these populations were migrating into. Um, how is it that like a mutation that helps protect against pathogens in the environment can also become like autoimmune and cause things like multiple sclerosis and rheumatoid arthritis?
Yeah, that's actually such a good question. I was like thinking about it this morning. And what I was like just thinking, I would say like a lot of these autoimmune disorders like MS and rheumatoid arthritis, because they can happen in later stages in life, and they're very like long-term diseases. Um, they usually happen after um the sexual like productive peak of a human's life. So these like mutations would have protected against the pathogens like some type of flu or some type of like virus. Um, but by the time someone who has this like has these mutations got got multiple sclerosis or survived long enough for like rheumatoid arthritis or something, they would have already had kids and passed that gene down. And the other thing is that the mutations, because of like the change, like I think it was just like a byproduct of the different changes the body or like our generations were trying to like pass down.
Thank you.
Yes. Are there any other comments?
I guess for me, like, I think this study is super interesting because usually they analyze like the molecular stuff, but not really like the ancestry. Do you think that there are other studies that do this as well? And like, is it beneficial?
Yeah, I think definitely for other studies, this one, interestingly, it's not actually in Nature Neuroscience, it's just in Nature because it's really more of like a genetic like analysis for ancestry. There's definitely like other studies that um look for correlations with some type of disease and then if ancestry or wherever like the genes like date to if they have like a correlation. And then for your second part about its use, I think it'll be very like helpful for us knowing for the people to protect themselves or like be able to predict if they'll have some type of autoimmune disorder later in life.
I think going off of Danny's question, I think you previously mentioned that like we can't really continue to do a lot of like the analysis that is done in this study because of like vaccinations and how they kind of like don't show the proper like ancestry. How do you think this is going to impact like future studies? And do you think in the future we might be able to like overcome this issue?
Oh, um, for future studies, I'm honestly not too sure like how that aspect is gonna look in the future, but I think like as people just continue to dig up more specimens or find more evidence, there's future studies will still be able to like go on and still like analyze a lot of the genetic information going on. Because actually, one thing really interesting is that the genomes that they matched with present day European genetic ancestry, they had some post medieval and medieval genomes, which were new information that came out pretty recently. So I think that will actually start like it'll just continue to come out more and more. And then as for, I'm sorry, what was your second question?
I forgot.
I think your second question was about,
I think how we can overcome this or how we can kind
oh,
of work around this.
yeah. I think there's certainly like a way to do that. Mm, there are a lot of like new analyses, like new forms of analyses like coming out. So I think like with that, we can definitely find more information. And then as like my answer from before, like with like the ever expanding like database, I think there will be limitations, but we'll find a way to help make the research useful to us in the present.
That was really insightful. Thank you.
Ah, thank you so much. All right. Um, so I guess that concludes our first Neuronic Book Nook. Yay! This is really
Yay.
fun. And then like just I love it so much. All right. I'll stop sharing and I'll stop the recording.